Hey all, Miss O'Leary here. I'm going to do a mini lecture to help you with some of the details and an overview of the epigenetic modifications that we have been talking about as a class. The first major epigenetic modification is DNA methylation. It's important to differentiate whether we're talking about DNA or histones with methylation, and that can be a little confusing. So right now we are focusing on modifications to the DNA itself. So if you look here on this picture up on the left, we can see that methylation happens specifically at cytosines. So of our ATCG bases, cytosines are the ones that get modified with methylation. So DNMT is our enzyme. This is DNA methyltransferase, and it is very often taking the methyl groups off of S-adenosyl methionine. And so this CH3 group is removed with the help of this enzyme, and then the CH3 group, the methyl group, is added to a cytosine. Okay, so it's just a little extra carbon group, but it can have a big difference. So you do have to remember that when we look at DNA, the sequence is what determines what proteins have affinity or bind in particular regions. So for example, if you look here on the right, this is RNA polymerase in the green and blue. The orange is the DNA, and the red is the RNA that's being transcribed. So you can see that the polymerase is sort of wrapped around the DNA. So imagine that that has to be true also where it first binds, so at the promoter. But if you have these methyl groups, so you can see in both pictures or down here, these methyl groups block the sequence. So remember, we're talking three-dimensional helix structure. These proteins have to basically recognize and bind to this chemical makeup. But the methyl groups change that. You can also think of them kind of like spikes. So if there's spikes in that region, then that protein can't actually bind there. So where you have methyl groups, it prevents the binding of proteins to that region. So in that sense, DNA methylation is pretty much always silencing. So it turns down or turns off expression in a particular area. You can get methylation in other places besides the promoter, but the same thing pretty much applies. So if you have methylation in regions where activators or repressors bind, the methylation will still repress binding. The outcome on transcription can be different based on what is supposed to be binding there, but the major cell differentiation related silencing events tend to be where the modifications are near the promoter. So again, modify the promoter, you shut down transcription, so that can essentially turn a gene off in that region. The next major modification is to do with histones. So you can see in this picture, histones are actually an octamer. So they have these H3, H4, H2A, H2B parts that all come together. So you get basically two of each set to make this octamer. And you can see that all these little scraggly tails hanging off of this. So it isn't like a full circle here. And those tails are what you see interacting with the DNA in this picture on the right. So the modifications that happen tend to be on the tails of these histones, which is where it interacts with the DNA. So the modifications can then impact that interaction. Now, DNA is overall a negative charge because of the phosphate groups in the sugar phosphate backbone. And a good way to attach to negative things is to be slightly positive. And so we see positive charges in different areas along these histone tails that help it interact with the DNA. So the positive charges on the histones can actually be blocked. So that is essentially what is happening with these acetylation modifications. So if you look down at this picture on the bottom, we have closed chromatin. So these are the histone octamers, and we have our DNA pretty tightly wrapped. However, if we use HAT, which is histone acetyltransferase, we can take the acetyl groups off of the acetyl-CoA molecule. And what happens is these acetyl groups are neutral. So if we attach these acetyl groups to positive amino acids on the histone tails, then it will neutralize the charge. And that means that it won't be as sticky, basically, for the DNA to attach to it. So there's going to be less attraction between the DNA and the histones because we've taken away the opposite charge. And so because of that, it loosens up the DNA. So instead of being, with the opposites attracting and being very tightly wrapped together, it will loosen up. And you can see up on this picture on the left, the benefit of having these loosened up is that proteins can bind in the areas of DNA that are now more accessible. So that can be RNA polymerase, it can be some of our basal transcription factors, or it can be different regulators. So histone acetylation pretty much always has the same outcome, that it can increase expression in areas that are acetylated because the DNA is looser. That being said, acetylation is not the only histone modification that can happen. So you can see in this picture, it looks really complicated, but up on the left here, this is our main histone octamer. And then you can see the tails coming off of it. So the H3, H2A, H2B, okay. But there are several different types of modifications. The little yellow AC, that's our acetyl group. So we already talked about those. But you can also see that there's phosphorylation. So P is phosphorylation. ME, this is methylation. So this is methylation of a protein, of the histone protein, not the DNA. That can occur in some places. And then actually sometimes you can get UBs for ubiquitin. So you can get little ubiquitin markers as modifications as well. So we know what the acetyl groups do. So if you look down at this second picture, everywhere we see these acetyl groups, you can see that this is, any histone acetylations are basically active marks. So that means the DNA in that area that's touching that histone is more likely to be active. You can see like this orange tail over here has just acetyl groups on it. However, you can see that this H3 tail has a combination of methylation, phosphorylation, and acetylation. And so some of these, these refer to the histones and their modifications down here. So H3K27, for example, will be up here. So this has a inhibitory or a repressive mark on it because of the methylation. So you can see that overall, you have to look at a combination of all of the modifications on the histone to determine whether the DNA in the area is more likely to be active or not. It's not just a case of acetylated or not. In reality, it's pretty complex with all the different things that can happen with these histone tails. All right, so one of the important things to research is to look at these together so they don't occur in isolation. So you have to kind of look at the combination of DNA modifications and histone modifications to determine predicted expression. So for example, in this picture, we have methylated DNA. So we know that methylated DNA prevents binding, and the histones have the DNA tightly wrapped around them. So the combination of not being acetylated and being methylated will help shut down expression in this area. So this is basically, there's two avenues to silencing, and it basically confirms that this really won't be read. So that would be important for things like differentiation. However, on the other hand, you can see if the DNA is not methylated and the histones are acetylated, then we get exposed DNA in between the histones, and there's nothing blocking the binding of the proteins. So it's accessible, and there are no little spikes or anything on it. And so we expect to see more transcription in this area. This is another picture of something very similar. I like this because it shows this is a nuclear lamina, so how your chromosomes would be interacting with the lamina. You can see that if there's active transcription, the shape of the chromatin is a little bit different in between these two pictures. So if it is acetylated, it's a little bit more open, and it's not methylated, then we see expression of RNA. However, if it's tightly wrapped, so it's not acetylated, and we see methylation in that area, it shuts down transcription effectively. All right, so the last epigenetic modification or regulatory scheme I'll talk about is microRNAs. So this is a rather thorough process, as you can see on the screen here, the steps of using microRNAs. But the focal points are at the top and the bottom. So we see polymerase 2, we're making an RNA. Okay, so we have basically your primary microRNA. Remember that RNAs can take three-dimensional shapes. And through this process of using dicer, and there's copies that are split apart here, we get this microRNA, so a short piece of RNA. So what happens is this RNA has a sequence that will match up this microRNA with part of a regular mRNA from transcription. So if this microRNA will bind, so if the sequence is similar enough and it binds to an RNA in the cytoplasm, for example, you get this little double-stranded piece of RNA. And in that case, this RNA will be chopped up. So the double-stranded RNA will basically tell the cell to cleave this structure apart. And that means that you shut down the translation of this RNA. So it's been transcribed, but because of the microRNAs, it will not be translated. You can also have these microRNAs that do direct inhibition of translation. So they might change the structure so that the ribosomes can't bind. And so again, you shut down the translation of this RNA, this mRNA into a protein. There are other sorts of short non-coding RNAs in the cell that can cause regulatory changes, and those are also important. I will say that a lot of times with microRNA, we see a correlation right now in a lot of research, and they're still looking at the causality. So we can say that certain cell lines have upregulation of really specific microRNAs, but one of the things that is being researched is what actually are they doing? What's the mechanism by which they're functioning to regulate the cells? And so again, this one will also be involved with crosstalk with the other two. So all of these epigenetic patterns together can play a cohesive part in how a particular area of DNA is transcribed. So hopefully that was helpful to clear up a few things and give you another overview. If you need to review epigenetics, please make sure to ask questions anytime that you have them.